Welcome to the Aging Boomers. I'm your host, Frank Sampson, and this show is a wonderful show. At least that's what I'm hearing from all of our listeners. And we discuss many of the issues facing boomers and their parents, and what we know is definitely an aging population today. We, of course, have a great guest today, but before I introduce Daniel, I'd like to just thank everybody for all their support on the show. We're getting a lot of recognition on iTunes, and you, of course, could go to iTunes and just do a search and subscribe to the show, The Aging Boomers, and get updated on all the upcoming shows, which is held every Friday morning and Monday morning Pacific time. And we will. You also can go on to your apps, either your on iPhone or on your Android phone, and just download the app, The Aging Boomers. So we're excited about that as well. So we have with us today Daniel Zwilling. He's a PhD and is with Circuit Therapeutics and part of a discovery team to find new ways of treating psychiatric disorders by using an innovative platform that allows the identification and characterization of drug targets from brain circuits involved in normal and pathological behaviors. Prior to his work at Circuit Therapeutics, Daniel was at the Gladstone Institute, where he used his extensive experience in behavioral paradigms of learning and memory, anxiety, and motor testing. Daniel has extensive experience in animal behavior, translational research, in vivo optogenics, we'll get the definition of that shortly, disease models of neurodegeneration, drug discovery, protein biochemistry, and molecular biology. He says he is determined to contribute to research in psychiatric disorders and neurodegenerative disease, including Alzheimer's disease and Parkinson's disease. Daniel, it's just an honor to have you on the show, and thanks for joining us.
Thank you so much, Frank, and thank you for having me on your show, and it's a pleasure to be here.
Yeah, well, first of all, I just want to thank you for all the work you do. There are millions and millions of people depending upon you and your peers to help hopefully find a cure to some of these, diseases, brain disorders. So thank you for all your hard work.
Oh, it's really my pleasure. It is fun to work in research. I've always been interested in research, and I can assure you that there's literally thousands of other researchers in the world who feel the same way and who are really working hard every day to try to find cures to the diseases that are that people are suffering from today.
Yeah, so I had to practice a couple times, even when I was introducing you, to pronounce all these all these words, and probably when I was doing it, I would say that a number of the people that are listening probably goes, "Whoa, what is he talking about?" So, could we define some of these things? And I think we're going to spend probably most of our time on an area called optogenics, which you work on today. Can you tell us a little bit about that and what's that all about?
Definitely. So it's optogenetics, which is basically a composite word of opto and genetics. So basically what this allows us to do is to modify neurons in a way where we can control their activity with a switch of a light. So optogenetics really represents a very new and promising approach to understanding and potentially treating neurological and neurodegenerative diseases. Optogenetics allows researchers to control the activity of brain cells with the use of light. And this is done by genetically introducing light-sensitive proteins into nerve cells. And this is allowing the scientists to control the activity of that nerve cell very precisely in space and in time. So basically what that means is we have an optical approach. We use light in order to activate a neuron or deactivate or silence a neuron. And we can do that in a petri dish in the lab where single neurons that have been cultured live. But during the past decade, we have advanced so much that we can now actually activate specific neurons in living animals in the lab.
It's amazing. So are you saying that scientists like yourself can turn these pathways in the brain on and off just like a light switch? Is that what you mean?
Well, yeah. Basically, I have a laser that I can use to switch a set of neurons or a brain region on or off in a animal. So that's exactly right. So what I can explain a little bit how it works in more detail. So what we're doing is we use proteins that are called opsins, and those opsins naturally occur in our eyes, for example, in the retina, where they respond to light. So when light is shining onto your eyes and onto the retina, these opsins react to the light, and they signal to the brain that you're seeing things. And some of the unicellular microorganisms that live in the oceans and on this planet, they also use opsins to move charged molecules, which are also called ions, across the cell membrane. And they use these opsins to produce energy. So they move ions across the membrane, and when the ions flow back, they use this flow of ions to produce energy and to live. So nerve cells, or what we call neurons, use the flow of ions across their membranes to produce electrical charges. So these electrical charges are produced, and when there's an electrical discharge, it creates a spike. And maybe you have seen, or some of your listeners have seen, when researchers visualize the activity of neurons as really little spikes that you see on the screen. And these spikes are essentially the language of the neurons, which are similar to our Morse code. They are little impulses that enables the neurons to communicate with each other. And so these nerve cell firings allow us to think, to remember things, to move, and to have emotions. So they're really the language of the brain and all the nerves in our body. So two researchers here at Stanford University, Carl Deisel-Ross and Ed Boyden, were among the first to realize that these opsins could be used to activate or inhibit the neurons at our will in the dish. And initial experiments used genetically modified neurons in the dish that contained the DNA for the production of these opsins that would activate the spikes. And so when shining light on these neurons, they would show spiking activity. And there's also other types of neurons that when you shine light on those opsins, you can deactivate the neurons. So depending on which type of opsins you use and how you alter the neuron in that particular experiment, you can either activate or deactivate a neuron.
So a neuron that spikes is
a neuron
one
that, spikes,
that
exactly.
you'd want to turn off?
I'm sorry?
Is a neuron that's, if I'm asking the question properly, one that spikes, so that tells you that that's a neuron you want to turn off, or am I not asking this in the right way?
No, that's exactly the right question. So when neurons spike, they talk to each other. But oftentimes, neurons may not spike enough where it's necessary. So you would want to activate those neurons and help them spike more. Other neurons maybe spike uncontrollably. And so when they spike too much, you want to shut them down a little bit and, push them in the other direction.
So give us an example, Daniel. Let's say with Alzheimer's,
right.
Give us an example of a neuron that you would want to either turn off or turn down, and another neuron that you would like to, increase.
Right. So, for example, in Alzheimer's disease, you have certain subsets of neurons that are degenerating. So your listeners may be familiar with the brain regions that are affected in Alzheimer's disease. One of them would be the cortex, and the other one would be the hippocampus. And the hippocampus is really a region that is important for learning and memory. The hippocampus is connected to another brain region that is called the septum. So in this case of the septum to hippocampus, you may want these neurons are one of the first neurons in Alzheimer's disease that degenerate. So one thing you would hope for is to have these neurons fire a little bit longer, as long as they're still able to fire, and help them do their spiking activity more, because these are really the ones that stop firing at some point, or where the firing goes awry, and you would want to increase their firing. Within the brain, there's also other neurons that they are actually inhibitory neurons that keep other neurons in check. So they actually turn other neurons down, and if they wouldn't work properly and are not properly doing their job, you would also want to increase their activity or turn the ones down that are firing too much, depending on the type of neuron you have and the task that they have within the brain. So we do know now, after decades and decades of research, that a neuron is not just firing and that's it. There's networks of neurons that control each other and that have specific spike firing patterns, and that are only active in certain points of time, and that really build a very difficult network amongst each other that has to be fine-tuned and stay that way. And if anything goes wrong in there, it could potentially lead to some of the dysfunctions that we see.
So would you say also that, I have interviewed people for a while now and do a little bit of reading on my own aside from that on the various diseases and what's going on, and there seems to be there's groups out there trying to prevent these diseases, and then there's groups where the concentration is more on a cure of these diseases. It seems like what you are doing and your team is doing is really trying to find the cure. Am I right?
So our approach is really two-pronged. On the one side, you can use optogenetics, and I think that's actually true for many of the research groups that are out there. On the one side, every researcher has to first try to characterize the system that they're working with. So we have to first understand how the brain works in a healthy state, and then understand what is going wrong when we have a certain type of disease. And then when we know what's going wrong, we can either try to prevent that from happening, or when the damage is already done, try to reverse the damage and get the brain back to a healthy state. And so in a way where, depending on where your what your strategy is in your research, you can either try to find drugs that prevent the degeneration of neurons that where you know they will probably degenerate in Alzheimer's disease or other diseases, or when those neurons are already degenerated and you have a patient who is already suffering from certain symptoms, try to find drugs or medical devices that will help the neurons cope with the situation and restore this imbalance in these circuits. In general, I would say it's probably easier to prevent damage than fixing it. If you have a car and it breaks down, it would have probably been easier to go to the regular maintenance and make sure it's running smoothly than sitting on the side of the road and trying to fix a motor that blew up on you. So in a way, prevention is probably better than trying to fix the problem in retrospect. But it is difficult in terms of neuroscience because sometimes we may know what the cause is of a certain dysfunction in the brain, but we may not be able to restore it to a healthy state because we don't have the drugs at hand or the molecules at hand or the technology to really do that.
So how do you go, what's the procedure for this? In other words, are you really opening the mice up and doing these procedures? How is it done? And we don't need to necessarily get too graphic, but I'm just trying to understand that part.
We're in fact doing brain surgery on the mice. So what we need to do is, and actually this is very similar of what's been done already in patients. And of course, before we go into the patients, everything is tested in animals, but you may be familiar with deep brain stimulation for Parkinson's disease. This is where you insert an electric probe into the brain of a patient to the brain region that is affected by Parkinson's disease, and this electric probe will then put a very minute amount of electricity onto the neurons in order to restore their functioning. And so what you need to do is you do brain surgery, you find the exact location that you can put your probe into the brain, and you close everything up, and people can literally walk around, and it's almost like a pacemaker, but for your brain. You don't see it from the outside, the people don't feel it, they only know that they're symptom-free. And so what we do with the mice right now in terms of our optogenetics is very similar. We are inserting optical probes that deliver laser light to the tip of an optical fiber into a specific brain region. And the mice are running around, they're grooming, they have their cage mates, and they're happy. We literally do brain surgery on the mice in order to modify these circuits in their brain.
And how long has this been going on? This research.
So this research started about 10 years ago. People, when I talked about the initial experiments that they did, those were, of course, experiments that were done in individual cells. So cells that were cultured in the dish, and so then people moved on to animals like flies and worms. Many geneticists work with flies and worms because they can be easily handled in the lab. And you have hundreds or thousands of worms in the dish that you can do genetical experiments with. And then they moved on and they started putting these opsins into mice, and there's a variety of different animal models out there now that help researchers find out how the brain works in those models.
All right, so I'm a boomer. All right, so you kind of have an idea of my age, but I'm 61 right now. And do you think that I will see in my lifetime, or that this is being tested on humans, and we're seeing some progress? I hate to put you on the spot, but because we have listeners out there who are going through this with loved ones. We have listeners who are concerned about their own well-being and their family's well-being in the future. So I hate to put you on the spot there, but what do you think?
I just wanted to clarify. So the optogenetics has not been tested on humans right now.
No, right.
Only the deep brain stimulation that is already used in patients is an approved procedure. The optogenetics is still far away from being put into humans, so we're not doing that at all. We're still working with lab animals here. Just to clarify this, so people don't think in next month they can go to the doctor and get the optogenetical approach. So this is really just in the preclinical phases. But I want to say, I have not met you in person yet, but from what I can tell from the pictures that I see on the net, you don't actually look like 61 at all. You more look like 51. So I think you're
well, 60
much younger.
is the new 50, they say, or something like that. Yeah.
And so let's say in this day and age, people can easily live to be 90 years old. Or even older, and so I guess you have another several decades to see what's going to be happening in research and the progress we're making. I cannot really predict what we will see in 40 years' time, and it's very difficult to predict this. I always think about 20 years ago, the use of cell phones was really not very common. And my first cell phone was maybe able to receive calls, and I was able to send text messages to someone. But even writing the text message would take me 15 minutes because it was so tedious. And today I pull out my iPhone, and I can see a satellite image of the street corner that I'm standing at. And had you told me 20 years ago that I would have an iPhone now that can do all these things, I would have probably said this is science fiction. And similarly, 20 years ago, I thought that only in sci-fi movies people would be able to control their spaceships with their thoughts. And I don't know if you saw the FIFA World Cup opening ceremony in Brazil last week.
Yes.
But there was actually a paraplegic person who did the first kick on a football by controlling an exoskeleton, which is a robotic scaffold that supported his body. And he kicked the ball by controlling the exoskeleton with his thoughts. So his brain activity was measured on the outside, and the robot executed his thoughts. I mean, had you told me this 20 years ago, I would have said, this is crazy, right? So we have come so far that it's really hard for me to say what's going to be in the next 20 and the next 40 and the next 60 years. It's crazy, and I think this is the right time now to actually ask these questions, because we're really in this revolution that's happening in neuroscience right now. We've seen the computer revolution. We've seen the biotechnology revolution, and people say right now we're in this age of the neuroscience revolution with things like optogenetics coming up and people controlling robots with their thought. I think there's going to be a lot of things that are going to be happening in the next couple of decades. And, let's call me up in 30 years and we chat again, and then we can talk about what wonderful new things.
Hey, if I'm still doing this podcast in 30 years from now, it will be my pleasure to call you up and talk about it.
I will be there.
Yeah, Daniel, just we have just a minute. Is there any thoughts that you have that you'd like to share with our listeners who may have loved ones with one of these disorders we spoke about, Alzheimer's, Parkinson's, even Huntington's, but just any thoughts you have that you'd like to share with them? We just have about another minute.
Right. So I would just like to tell your audience that they can really rest assured that there's literally thousands of dedicated researchers in the world who are working very hard to find a cure for neurodegenerative disease and psychiatric disease. And many of them have really dedicated their life to research. I know professors, and researchers in the university and in the companies that really work day and night and for 30, 40, 50 years. Their careers are really based around their research. And they're doing everything they can, and they're also the smartest people on the planet, and they're doing everything they can to try to find something. And I would really like to say, never give up hope that we will one day find something that can help. And in addition, it's probably also important to know that nobody has to go through this alone, and that there are many very good resources out there, including web pages of the Alzheimer's Association and the Parkinson's Association and the Huntington's Disease Society of America and things like that. Plus, we have people like you, Frank, who are dedicated to make a difference in people's lives by advocating for them. So I think altogether there's good resources, there's people who can help with the daily challenges, and don't give up hope that maybe in the next few years to decades to at some point we will find something, and modern medicine has done a lot of progress in many areas. If you think about other diseases that may have been devastating 50 years ago, and now we can control them. I think there's good reason to think that for neuroscience and neurodegenerative diseases, we will also find stuff in the future that will help.
Daniel, it was a pleasure having you. If people want to do a little more research themselves on optogenetics and what you're doing, where would they go to do that?
So what they can do is really just type in if they are online, have internet, type in optogenetics. Google will definitely find interesting news articles. Optogenetics has been described in a variety of magazines, and even the New York Times had several features about the progress of the optogenetics. I think that would be the best way to go. There's also some, maybe YouTube videos that people can Google and see how it is working with like animated videos and stuff like that. Universities are usually very good in putting out there what they're doing and how the technology works. And so I think that would be the best way to go.
Great, thank you, Daniel. Daniel Zwilling, PhD with Circuit Therapeutics, and thank you again for all you do, and we'll have to have you back and get an update.
Definitely, Frank. Thank you so much for having me. It was a pleasure.
Thank you, and thanks to everybody who's joined us today, and go to the Aging Boomers on iTunes, download it on your iPhone or your what's the other phone? Android phone, of course. Sorry, Google. Thanks for joining us. Be safe out there, and we'll talk to you all soon.